Can everybody hear me all right? Perfect. So tonight, we're going to talk about one of the many projects that I help research with my incredibly dedicated team here in the Florida Keys. And it's going to be Hogfish Ecology in the Dry Tortugas. We already had the show of hands of who here has been to the Dry Tortugas. I also raised my hand. I have been there. And it is one of my favorite places in all of the Florida to dive. It's truly unique, so if you haven't had the chance, strongly recommend. So who am I? I am actually the lead of the Finfish Research Team here in the Florida Keys. So my team does all of the finfish monitoring throughout the Florida Keys, from all the way up to Biscayne, out to the Dry Tortugas. And when I say finfish, I mean we are not focusing on elasmobranchs. So we are not sharks and skates. We are more teleosts, more bony fish. So snappers, groupers, everything down to the blennies and the gobies. So my team does all of the research monitoring there to help inform management decisions for the Florida Keys Fish and Wildlife Conservation Commission. So is everybody familiar with Florida Keys Fish and Wildlife, or Florida Fish and Wildlife Conservation Commission? So you know that we have a branch of law enforcement. Everybody knows our law enforcement people. But we also have a branch that's called the Fish and Wildlife Research Institute, and that's where I fit in with my incredibly dedicated team. So these are all of the people that help me do the research that I'm presenting here tonight. And I want to give them strong kudos, because none of this is possible without teamwork. So we are located in a field station in the Keys. Our field station for FWC is located in Marathon. Did anybody know we had a field station in the Keys? Okay, less than the people who went to the Tortugas, cool. So we have a field station in Marathon, and like I said, we cover from about Biscayne all the way out to the Dry Tortugas with our monitoring. And on any given day, we can do a lot of different things. So there are times where we are in meetings with our commissioners presenting research to help make management decisions. There are times where we are seining in seagrass beds to look at juvenile abundances. We are diving to count reef fish. We are conducting reef fish visual census surveys with a lot of other partner agencies. That happens every other year in the Keys, where we do hundreds of sites throughout the Keys, counting fish on the 4 reef area. We also can sometimes be in places that aren't even shown here, like up in the air doing aerial surveys to look at boating pressure. So we have a wide variety of activities that we could be doing on any given day. But one of the things that I find the most rewarding is the fact that we can present our data and have it be used in management decisions. So we can point to management decisions here in the Keys and say, our research helped shape that decision. And that, to me personally, is one of the things I find the most rewarding about my particular position with FWC. So for example, the research that I'm presenting today is actually being fed into something called the CDAR assessment for hogfish, which is ongoing right now. Does anybody here know what a CDAR is? Or a stock assessment? So a CDAR is the Southeast blah, blah, blah assessment research team kind of thing. But basically, this CDAR is how we do a stock assessment. And right now, hogfish are under review for the next stock assessment. So a lot of the research that we did in the Dry Tortugas fed directly into the data for this management rethink. Now, as I said, today I'm going to talk about a hogfish and the Dry Tortugas. And some of you already know how awesome hogfish are, but for those of you who might not be on the same bandwagon as me, I want to just explain why hogfish are awesome and why we were interested in studying them. So hogfish are the largest member of the wrasse species. So if you hear them called a hogfish snapper, that is incorrect. They are a wrasse. The largest record for them came from South Carolina. So they come from South America all the way up to the Carolinas is their range. And it was 21 pounds, 15 ounces. Yeah, caught in 2011. They have a larval duration of about 34 days. You can move a lot of ground in the ocean in 34 days from the time you are hatched out of your egg all the way to settlement. They typically settle in nursery habitats before they then move onto the reef as adults. They're considered generalists, and some people say that the way they got their name hogfish is because the males get a protracted snout and they root around in the sand looking for their prey items, which can be anything from polychaetes to mollusks. They're generalists. And one of the things that I think is the most interesting is that they are sequential hermaphrodites. And it's socially mediated, is what we think. So sequential hermaphrodites, they are all born girls. Not a single hogfish is born a boy. They are all born as females. And then as they age, they will all eventually, if they live long enough, turn into a male. But it's socially mediated because if there is a male already present and he's holding a harem together, those females might not change right away. If that male is removed, a female might change sooner than she would have otherwise. So there's some social aspects to their sex change, but they are a hermaphroditic species, and the males can hold a harem of up to 15 females. And they have a protracted breeding season, so breeding can start as early as October and last through the spring. The only months that they haven't really seen breeding is dead of summer. This species also is what we say suspected of high site fidelity. There's a lot of published literature about the home ranges of hogfish. That's where part of this study came in. But the few studies that there were, two, suggested they have a very small home range, but they weren't tracked for very long. So we didn't know if they had a small home range all year long, only during breeding season, only outside of breeding season. There were still a lot of questions about their movement behaviors. And then also this fish is historically subject to intense fishing pressure, mostly by spearfishing, but hook and line has actually become a little more popular for catching hogfish as well. And so this animal was in the Keys, was overfished and considered subject to overfishing still. And so that's part of the management changes that have been happening, is to try and bring hogfish back to previous levels. So in 2017, management did change for hogfish, especially in the Southeast Florida region. So in this map over here, the pink area is what we are calling the Atlantic waters for hogfish, according to management, and the blue hash lines are what they consider Gulf waters for hogfish management purposes. And in the Gulf, the minimum bag size is 14, or minimum size limit, sorry, is 14 inches and five per person bag limit year round. In the Atlantic, it's 16 inches and 1 per person, and there's a closure. And the closure is supposed to coordinate with the peak of their spawning season. If you want specifically all of the regulations, that's what the QR code will take you to, the FWC page where the specific regulations are laid out for hogfish. So these changes went into place in 2017. And like I said, right now, we started the reassessment of hogfish for management purposes. We actually started this in August of last year. And it's ongoing. It takes usually about a year to push through all of the data and the modeling. Yeah.
Any difference between spearing and...
Depends on your skill level. In the rules, no. You still have to hit certain minimum sizes, and yeah, in those dates. Hogfish are also very interesting to a lot of other people for lots of other different reasons, not just because they're sequential hermaphrodites, not just because they can get to be the largest member of the wrasse species, but also one of the things that I find pretty fascinating is their ability to color change. And if any of you have stared at them underwater, you know what I'm talking about. They actually have proteins under their chromatophores in their skin that actually takes, they call it like taking a Polaroid of their skin to help match their surroundings, so that this process of changing color doesn't even have to go through their eyes to their brain. Like their skin can do it. And if you want to read more about that research, that's that QR code at the top. It sends you to an article that summarizes the paper that did this research, the people that did this research about their color changing abilities. And they're so awesome that in 1926, when people were pioneering underwater color photography for the first time, the very first underwater color photograph was taken in the Dry Tortugas of a hogfish. So come full circle. So you're hooked, right? You're like, yes, I understand that hogfish are awesome. Now, why did we pick the Dry Tortugas for our study? And the reason we picked the Dry Tortugas, one, it's pretty awesome in and of itself, but two is because of the management zones in the Dry Tortugas. So Florida Fish and Wildlife teamed up with the Park Service to conduct this study specifically in a region of Dry Tortugas National Park known as the Research Natural Area, or the Research Natural Zone. And this zone, which is right here, It's 46 square miles, and it is completely protected. No anchoring, no fishing, and it's been protected since 2007. So in theory, there are potentially multiple generations of hogfish that have never experienced fishing pressure at all inside this zone. And if you want to set a baseline for age and growth, how quickly things can change for these animals in other parts of the Keys with these fishing pressures and with changes in management, you want to know, well, what does an unfished population of hogfish look like? How do they act? How do they move? What do they do? How do they grow? And so that is where we started with this study, was coming up with this information to then be able to compare it to other areas that are still experiencing fishing pressure. And that is what I'm going to present to you tonight, because now you are all hooked, right? You're sold on hogfish, and you're sold on the Dry Tortugas, and you're like, this is great. So buckle in, because it's gonna get wild. So this research had a two-pronged approach. The first part was biosampling. We were permitted to take up to 100 hogfish from the research natural area, and we were going to sample these fish heavily. So we were like, if we're allowed to take these fish from this very special place, we are gonna maximize every square inch of these fish. So we took tissue samples from almost every part of this fish, and we are working with partners still to get some of that information processed. For example, we are working right now with a postdoc who is helping us with the genetics. So some of the sampling is going to her research, looking at hogfish genetics throughout the region as well. The second part was acoustic telemetry. Has anybody here ever heard of acoustic telemetry? Yeah, okay. Acoustic telemetry, I'll explain it more later, but basically this is how we tagged fish and watched where they went. And we were able to acoustically tag 28 fish inside that area with no fishing pressure. And we used barrier nets and underwater tagging methods. So we tagged the fish underwater to reduce barotrauma to increase survivorship, because nobody had internally tagged these fish before. And we had heard from other colleagues in other parts of the state that they are susceptible to barotrauma when being handled for other reasons. So to increase their survivorship, we did everything underwater. And this map is showing you where we worked. So this is the research natural area, that 46 square miles of no take, no anchoring. Everywhere you see a teal dot, that's where we collected animals for the biosampling. Our acoustic array is inside that black circle, and that's where we tagged all of the fish that you'll see for the movement portion of this study. So as I mentioned, yes?
Question on the influence of lionfish in this area.
Question of the influence of lionfish in the, I mean, there's lionfish sadly in a lot of places, but there's not as many that I saw in our study area while we were out there. You're right, but like I said, in this area, we didn't see that many. We saw some, but not that many. This also is a shallower water system, and some people do hypothesize that lionfish come up from deeper waters and recruit in certain areas, and there's not deeper waters nearby. It might have absolutely nothing to do with it, but it might. No worries. So as I mentioned, we were permitted to collect 100. We only collected 97. Our last research mission, unfortunately, had poor visibility, which decreased our ability to catch we weren't skilled enough. But with these 97 fish, we extracted otoliths. Everybody here knows what an otolith is? Inner ear bone of a fish for aging them. We removed their reproductive organs. We also collected tissue samples from various parts of the fish. We also collected fin clips for genetics, and we collected stomachs. Some of this we had funding to process right away. Some of it we're archiving until we find additional funding sources to continue to process these samples. But like I said, this was a golden opportunity. We did not want to waste any part of these animals, and so we sampled as much as we could. So let's talk about otoliths. For those of you who are not familiar, they're the inner ear bone of the fish, and they're used for aging. So this is an actual hogfish otolith that is much larger than the actual otolith. They are more like a grain of rice inside the fish's brain cavity. And when you pull them out, you section them, and you will see rings. And the rings correspond to growth. Now, different fish grow at different rates, so you do have to have this validated per species. So what works for one species might not always work for another, but for hogfish, one layer of growth is typically 1 year. So if you see the opaque zone, that's going to be slower growth. If you see the translucent zone, that's faster growth. But one whole band is roughly 1 year. And we aged the hogfish that we collected. So this is an example of a sectioned otolith from one of the hogfish, Hogfish 73, that we collected. It was identified to be a male. This is the measurements. And then we collected it in March of 2023. And this animal we aged by counting these zones to be 10 years old. Now we say a margin of 1. Margins just add, when people say, oh, I'm 10 and a half. If it had a wider margin, it would be closer to 11. But with a margin of 1, it means there's very little margin available to be counted. So we're going to say this animal is closer to 10. And what does this look like for all of our hogfish? Well, all of the hogfish that we collected in the Dry Tortugas were between 1 and 12 years old. This is with a mean of a little under 5. The size range for the animals that we collected, I did put in inches here. I forgot it on the last slide, but I did put it in here. The size ranges were between just under 8 inches all the way up to just shy of 24 inches for these hogfish. And what I did here in this graphic is you'll see the ages on the x-axis and their fork length on the y-axis. And then we fitted it with something called the von Bertalanffy growth curve. And this is a growth model, basically predicting out, if you came back to this model, you could say, I have an animal of this size, and it could tell you, okay, it's probably this age. Or vice versa, I have an animal of this age, okay, it's probably this size. So this is fitted to our Dry Tortugas fish. So the oldest fish we found was 12 years old. The youngest one was 1. And these were the size ranges of the animals and the distribution of their ages. Now, how does this meet up with previous hogfish studies? And that's what we're going to talk about right here. Most of the life history work done on hogfish in the region was done a very long time ago. That was another reason why we wanted to do some of this work now. McBride and Richardson published their work in 2007, but the hogfish were collected as early as the 1990s that they used in this work. They aged over 1,000 hogfish. 1,400 plus hogfish were aged for this study. They collected most of these animals with cooperation of fishers, both commercial and recreational, and direct sampling themselves. This is a map from their paper of where the samples came from. And they divided it into 2 groups. They divided it in, so you have circles for the Dry Tortugas and the Florida Keys, and then you have the diamonds for the West Florida Shelf, which they called the Eastern Gulf in their paper. This is a figure from their paper as well of all of the hogfish stages of growth and what those otoliths looked like during those different years. They found the oldest fish in their study was 23 years old on the West Florida Shelf. The oldest Dry Tortugas fish they found was 13, and the oldest fish for the rest of the Keys they found was 12. This, by the way, is their 23-year-old fish. That's the otolith from their 23-year-old fish. So if we look at our age and growth curve with their age and growth curves, we can kind of see where we fall out. So the blue solid line is our Dry Tortugas fish that we just collected. And then these previous lines are the different areas from that 2007 work that was published. And so as you can see from here, now this is preliminary data and I have not run confidence intervals on all of these yet. So as you can see though, South Florida has a slower growth rate than our Dry Tortugas fish or the Eastern Gulf fish, which is this dotted line here. Now I cut this off at 14 because that's roughly the age range for the fish that we had collected, but this pink dotted line would go out to their 23-year-old fish. So you can understand that our Dry Tortugas fish towards the end doesn't seem to be growing quite the same way as those West Florida Shelf fish. Now, some theories about this, and one of them came from work done by Angela Collins and McBride in 2011. And they focused only on the west coast of Florida, and they found that older fish were in deeper water. And what they considered near shore and offshore, the 30-meter mark, or roughly the 100-foot depth, was kind of that break point for them between near shore and offshore. And they found that near shore, it was younger fish. Offshore, it was older fish. Another person, Ian Town, he was a grad student at Nova Southeastern, and in 2018, his thesis was looking at hogfish off of Broward County, specifically, a couple other places as well, but the hogfish off of Broward County that they caught, they found something similar. The older fish were in the deeper water, and the younger fish were in the shallower water, but he noticed that the younger fish grew a little faster, and they think that might be due to fishing pressure, that the fishing pressure caused those fish to want to grow a little faster, mature a little faster, get things done a little faster, and that the older fish in the deeper water didn't have the same fishing pressure. So they didn't have the need to grow quite as quickly. Now, we also did other types of sampling, like I mentioned, and one of the ones I want to talk about is the reproductive organs of the fish that we collected. So this is still ongoing. We collected 97. We only have 77 of the fish processed right now. We still have another 20 to go. But I can talk to you about the 77 that we do have. So we worked these fish up at the dock at Dry Tortugas, if you've ever been there. So right where the ferry pulls in, there's those big fish tables, and this is where we would work up our fish, and people would ask us questions, and we would answer them to the best of our abilities. And some people got very interested in it, and other people didn't understand that this is how you fillet a fish. So it was a very interesting dichotomy of people. But I think everybody walked away a little more educated. We removed the gonads. So the gonads are the reproductive organs. So we removed testes from the males, ovaries from the females, and transitioning from the transitioning. And this is what we found. We found that our hogfish had similar reproductive system, they fell into similar reproductive classes as the work from McBride and Johnson. McBride and Johnson, out of those 1,000+ fish that they had collected for the aging, they also got ovaries from 1,100 of them. So not quite all of them, but they got gonads from a lot of their fish. And so they made this model of the life history of a hogfish. So the first maturation is going to be as a female, and they're going to go through spawning, regressing, coming back into spawning season, and they'll do this until they start changing into a male. And they discovered that this change happens typically after spawning season. They typically mature into a male after a spawning season is done, and it takes several months. So if you remove a male from a harem during the middle of spawning season, we still don't know what happens. Does that female hold, the next dominant female, hold that harem together as she transitions? Do the females scatter? We still don't know the answers to those questions, but that's some of the things that they were asking as they looked at these gonads. So in this diagram from their paper, they go through all the stages of the female gonad, and then I have the same thing on the next slide for all the stages of the male gonads. And you can even see transitioning, this is female tissue, transitioning into what ends up being male tissue. And the transition typically happens from the outside of the gonad and works its way in. And it can take about 3 months, más o menos. So out of our 77 fish, 59 of our fish were females, and we found every stage of reproduction in these females. So young and immature or regressing, we found that, but we also found developing, we found actively spawning, but we didn't find them evenly distributed. We found them more heavily spawning, shockingly, during the peak of their spawning season. And then the regression was more towards the summertime. So it lined up very well. We did find 15 males, and we found 3 transitioning. And so if you look here, you'll see this is the female tissue, but you can start to see the male tissue growing in from the sides of the gonad. And then it'll continue to grow inward until you have very little female tissue left. This is actually probably a functional male at this point in time. So it depends, and that's going to bring me to this slide. Bam. So how does our data match up with data that was previously collected? So if you look, I have the Johnson and McBride paper here, and I have our preliminary study from the Dry Tortugas on that side. Oldest female in years, Johnson and McBride found was 12. We found 7. The youngest functional male they found was 3. The youngest functional male we have so far, we still have 20 fish to go, was 6. Ages for transitioning, they said between 3 and 5 years, and our 3 males were aged at 5, or transitioning males were aged at 5, 7, and 7. So if you take this with a grain of salt, because the caveat is there's a large difference in the sample size. If you remember, I said they looked at over 1,000 fish, and we looked at just under 100. So this is where we landed, but you have to take it with a grain of salt. So there are females that will mature by age 2, and if they start transitioning this quickly, they only may get 1 breeding season as a female before they start becoming a male. But if you stay a female until you're 12, you can get 11-ish breeding seasons from that female. Now, any questions on this part before I move to the acoustic telemetry part? Okay, so then let's talk about the second type of sampling, which is acoustic telemetry. Now, acoustic telemetry is where we internally tagged those fish with a transmitter, and this transmitter pings a unique ID code.
I may have missed it, but can you tell if they ever?
As far as we have seen, once they make the change, nobody goes back. It's a very straight line for them. They start out as a female. There's none that are ever born a male. They are all female, and then once they transition, we've never seen anybody with evidence of going the other way for their gonads. I don't want to say it'll never happen because nature proves us wrong all the time, but it has not been documented yet. Yes, it hasn't been documented yet, and we have, Richardson and McBride's work was over 1,000 fish, and they didn't see it either. So we feel pretty confident that it's.
Unless they're environmental.
I'm not saying nothing can happen, but this seems to be their life pattern. And if you have a female that can stay a female until it's 12, that social mediation for that sex change also seems to be a bigger factor right now than environmental factors. So if the male is present, they will stay female longer, older. And if the males are removed, there's evidence that they'll change quicker, but we've not seen anything go in the reverse direction. Any other questions? All right, so acoustic telemetry. Acoustic telemetry is where we insert a transmitter into the abdomen of the fish. This transmits a unique ID code for that animal. And we have our transmitters, in this particular instance, were set to ping once every 5 minutes, and they would last several years with the size of the tag and the battery that was in the tag before they run out of battery life. Now, acoustic telemetry has 2 parts. One part is tagging the fish, the other part is listening for those detections. And the way we did that is we set up these acoustic receiver stands. You put a piece of equipment in here known as a receiver. It has a hydrophone at the top, and it listens at the specific frequency of these tags. So it is always listening at that specific frequency of 69 kilohertz for these tags to ping. If a tagged animal swims near these, it will record a date and time of when that animal was present. Now, this particular study was unique for a couple of reasons. Like I said, we tagged underwater and the design of our array. First, I have a video that unfortunately also has my voice in it, but it talks about this project and it shows really cool footage of how we tagged these animals. And so let's play that and then we'll talk a little bit more. There we go.
Scientists from the Florida Fish and Wildlife Conservation Commission partnered with Dry Tortugas National Park to acoustically track the movement patterns of this species, hogfish, in a no-take marine reserve within the park. To do this, scientists captured hogfish underwater using nets. They inserted a uniquely identifiable acoustic transmitter into the abdominal cavity of the fish. To track these tagged fish, scientists deployed an array of acoustic underwater receivers that can record when one of these specially tagged hogfish swims by. Divers retrieve these acoustic receivers multiple times a year and download the data. With this information, scientists can document the home ranges of the hogfish, understand their habitat preferences, and can make better management decisions about these animals in the future. To learn more about hogfish research or other acoustic telemetry projects, please visit our website at myfwc.com.
It was very exciting, wasn't it? No, that's my colleague. I was the one videoing. So I'm very grateful to all of the people that help, and so I like to highlight as many of them as I can. to acknowledge the work and effort that they've put in.
So when the fish, does it identify?
Yes, of course. When the animal swims by, you ask, does it just identify a fish swam by versus an individual fish? And each of these acoustic tags has a specific ID number that it transmits, so we'll know which fish swam by. When we're doing the underwater tagging, we actually have a data sheet with us and we write down the serial numbers of the tags. And so we know that tag went into that fish. And then that's how we will be able to keep track of who went where based on where we tagged them. Yes?
If the fish gets fed,
That's an excellent question. Give me one hot moment. Oops, there we go. So I will circle back to that in just a moment. So just to reorient us, here's our map again. The teal dots are where we had animals that we biosampled. This black oval is where we set up our array and tagged our fish and watched their movements. And our array looked like this inside that oval. Now there's two different types of acoustic receivers here. And that's what I want to explain, and then this will help us with predation events. If you see purple dots or pink dots, depending on how you view color, these are going to be plain acoustic receivers that all they do is give you a date timestamp. They just say, on this date, at this time, this ID swam by. If you look at the orange dots, what you'll see are receivers that do slightly more. And these receivers, we call this a positioning system, and they actually can help us pinpoint exactly where the fish is within that array. And the way they do it is they are not only listening for the fish swimming by at that specific frequency, but they are also once every 10 minutes transmitting their own ID out to all the receivers around it. And by understanding the millisecond timing between when that information was received by another receiver and then the information of when that tag was received by both of them, that difference in timing tells us if it was closer to one receiver or another. And if you have a bunch of them together, you can tell when it swam closer to one receiver versus another and get within meters the location of that fish. They call it a hypotenuse as opposed to triangulating. So when I talk about it, yes, it's like triangulation. And that helps us understand, usually sometimes within a meter. But it's complicated a little bit by the environment in the Dry Tortugas. So if you looked at this, remember this image from before, this is a very flat environment. Sound travels really well. There's nothing obscuring the sound reaching a receiver. There's no coral head, there's no rugosity, there's no echo. We call it acoustic shadowing, where it could bounce off of something and not reach the receiver. In the Dry Tortugas, this is not always the case. And so if we go back to that receiver map, most studies can get away with placing their receivers in these positioning systems over 400 meters away. We had to bring them in to be closer to 90 meters to ensure that we had good coverage. So it meant that we had to put a lot more out to cover the same area as if this was in a less complex environment. And it makes for handling the data on the back end a little tricky as well. But one of the really cool things about this is when you do it this way, you get more information that is richer, and you can look at things like predation events. So if you are looking at data here, it's not going to tell you where that fish was in that cone that it could detect. It has a detection radius. It listens in all directions at the same time. So if this has a detection radius of 200 meters, it could be anywhere in that 200 meters around this receiver. So these are mostly just for presence absence. We just wanted to know if it left this area, did it go south? Did it go west? Did it go east? And that was all the information we were going to get from here. This will give you much more rich information, like I said, about behavior. And also, if the animal changes behavior suddenly, that's usually when we go, oh, normally they do this, and all of a sudden, they booked it out of here at a much higher speed than I've ever seen them go. That is probably a predation event. And so we can look at the data that way. And there are some that we don't know. Sometimes they exit the system and we never hear from them again, and we don't know why they exited the system. Was it because of a predation event? Was it because somebody was poaching in the Dry Tortugas and they weren't supposed to? We don't always know. But this richer data can help us answer some of those questions when we see an obvious pattern. So
they always, like, because of the nature of a hogfish wanting to stay in a certain area.
well, we'll talk about that. So, like I said, we set this out, and this was the discussion about the simpler habitat versus the more complex habitat. So what did we find? Well, we tagged the 28 fish. We did not tag them all at the same time. They were tagged over the course of a year. So at any given moment in our system, we did not have all 28 fish. You'd have maybe 17 fish, or maybe you'd have 22 fish, but we never had all 28 at the exact same moment. This is the size range of the animals that we tagged, and what we found was, yes, they love staying in the same spot all the time. So these are 3 hogfish, and these are all of their detections between April to that September. And I picked 1 male, 1 female, and 1 that we think is transitional, but unfortunately you can't verify that easily. It's just based on the physical observation of the animal. So it could be a female on her way to transition, or it could still be a functional female or a functional male. We're not quite sure. No, not always. So we're trying to do this as quickly as possible. So this is only based on coloration, external observations. So if you saw those terminal male fish, they'll have that distinctive longer snout, the much darker mask, they'll have the little spot on their side, and they act a little bit differently. So the males hold the territory, and so they'll patrol their territory every day, and they'll come up to you to threaten you, basically, and be like, get out of my territory. But to do that, they turn to their side and they shake at you a little bit, and that makes them an easy target for people to shoot and eat. And in their minds, they're defending their territory and they're showing how tough and big they are. They will do similar things to other males that are coming too close to their territory. They'll have little mouth fights, or they'll show off and they'll shake at each other, but they also shake at each other and flare those 3 dorsal spines at the females to be like, hey lady, check out my dancing. So they'll do that too throughout the course of their day. And when they're holding a harem and it's breeding season, some estimate that the males can breed 50 times a day with various females in their harem, which is a lot. Whew. Nope, I'm still good on my side. Oh no. Oh, okay. I love that. Oh, I'm not going to touch it. We're gonna go with that. But we did find that during most of our study, these animals stayed put pretty well from where we tagged them. And did it go? No, it did not go. Okay, let's try it that way. Well, it's going on my end, but it is not changing on this end. Okay. I'm going to unplug and plug back in. Yes? Yeah, now a word from our sponsor. All right, let's try this again. So, most of the time they stayed put, and most of the time they hung out in the same area, with the exception of not the next slide.
Like the advancer is not going.
Let me try it with this. It's advancing here,
I know, I see that.
but it is not advancing up here.
I think it's something with the laptop.
We did unplug and replug.
It's advancing up here, but not on the display.
Dun, dun, dun. Yes, now a word from our sponsor. Oh, there we go. That's good. Woo! So this is an example of a different time frame, but for fish as well. And you can see it's almost the exact same place. It's almost the exact same area. And like I said, most of the time they hung out here. There was one notable exception, and that is some of these animals we were able to get pressure tags into. And they're much more expensive, so we did not have the funding to do all of them with pressure tags. But some of them we were able to do pressure tags, and it was a pressure tag / accelerometer. So every 5 minutes it would send out a pressure and a location, and every 5 minutes after that it would send out an accelerometer reading, which is a rough indication of activity levels, and the receiver would tell us where it was. This is the depth reading profile from fish 15407, which was tagged in 2021. And if you look, this is the depth in meters where it normally hung out, and how many different number of observations we observed. So most of the time, it was right around that 7-meter depth. However, if you look way over here, there's some at the 21-meter-ish line, and we were like, whoa, what's going on here? And then we remembered this guy. And in 2022, Hurricane Ian not only came over the Dry Tortugas, but it came right over our array. And so the eye of the storm passed right over Garden Key, right over our array, and kept going. And we were not able to get back out there until December. So we had no idea the fate of our array, the fate of our fish, until we got back out there several months later to check on everything. The first thing we noticed was that our array might have moved a little bit, but surprisingly, a lot of them stayed put, especially in such shallow water. The top of these reef features are only like 15 feet deep. And the slope at the top up here is about 60 feet. It slopes down to about 60 feet at the top. And if you look, it doesn't seem like a good rhyme or reason why some moved and some stayed, and they moved in different directions and at different distances. The furthest one was this station, ended up all the way over here. It's part of our permit was we're not allowed to permanently anchor anything, but we tried to make them heavy enough that they would resist most things. Yeah, and it worked for a lot of these. It worked for a lot of them. They survived Hurricane Ian, but some of them moved around.
I'm not clear about the pressure sensor. It records the pressure at the time it sees the fish, or
It's the tag inside the fish, and it's recording the pressure that the fish is experiencing at that moment in time, and it's transmitting it out with the ID number. So it's the depth of the fish at the time.
The 20 meters, the sensors aren't recording that. There's those up there. So it moves.
Oh, you're taking all my thunder.
We need to know when we ask questions.
I apologize.
And we'll use the microphone.
I apologize. So he was asking questions about the pressure sensor and how they worked, and I said that the pressure sensor basically is recording the pressure of the fish at the time that the transmitted ID is happening. So it's the depth of that fish when that ID is transmitted out. And then he said, but most of your array is not at 21 meters. And I said, yes, you are right, except for the ones up here. And he said, oh, they moved. And I said, oh, you stole my thunder. And now we're all caught up. So if you look at the readout of this one particular fish that had a pressure sensor in it, this is time right around the time of Hurricane Ian. This is the depth in meters. And this circle is right when the hurricane was heading right towards the Dry Tortugas. And you can see that the animal had a normal daily habit of going up and down, and then booked it into deeper water, and then shortly after the hurricane came back and started resuming normal up and down patterns again. And if you remember, I said that accelerometer was on this fish as well, and this is the accelerometer data from this fish from around the same time frame. So normal pattern, and then it hunkers down. And so on this graph, the way you read it is the y-axis, the higher the number, the more active the fish is. The lower the number, the less active the fish is. And this is a relative, it takes a reading of the x, y, and z axis of the tag. So it's a relative number of activity. But you can see that during the hurricane, it hunkers down as best it can, but maybe it got blown out a little bit as it was trying to move around, and it was still a little hairy out there. And what I'm going to show you next is called an abacus plot, and it's of all the animals that we had in the system when Hurricane Ian hit, and it's going to take a moment for me to explain. but it's still pretty impressive. So these are all tagged fish. And these animals are the animals that had both a pressure sensor and an accelerometer. So this is actually one fish, but it has two different IDs because one's the pressure and one's the accelerometer. And this is the time on the x-axis, and this is every detection of those fish on this particular station leading up to Hurricane Ian, which is in that aqua oval. And if you look, a lot of these fish have very regular patterns around this station. Hurricane Ian happens, and a lot of them disappear for a day or so, and then many of them come back and resume their normal patterns around the station. So at the beginning, we had 15 hogfish before Hurricane Ian, but we only detected 11 afterwards. Some of them took a while to return. So if you look here, this is October 1st, this is November 1st. This fish was gone for a month, but eventually it came back. And then other fish, like this one, disappeared, and we never heard from again. And we have no idea. And we never heard from it, so we don't know if it hunkered down. We don't know what its accelerometer was saying or its depth patterns. It just, when it disappeared, it was gone from the array, and it never came back. So the fish that did come back, some of them took a little while, but a lot of them resumed similar patterns of behavior, but one of them did not. And he actually shifted his home range after the storm. He was still in our array, but not on this particular receiver the same way. He moved to it, or she moved to a different receiver, and was still in our system, but had shifted just a little bit its home range. So with this, we have some ideas of what's next for the movement data. And one of the things is looking at individual variation. What patterns might emerge from the age of the fish? Like, do certain ages have certain home ranges, or certain sizes have certain home ranges? Do the males have a different home range than the females? Also, what are drivers that influence the movement? Obviously, Hurricane Ian was one of them, but are there other influences on these behaviors? So if the temperature increases, does their home range expand or contract? If the lunar phase, for some animals, as you know with Grouper Moon project, the lunar phase is really important. Might not be that important for hogfish because they have such a protracted spawning season, but maybe there's a signal. Is it more linked to breeding? So during the spawning season, is their home range a certain size, and outside of spawning season, it's a different size? So these are all of the questions that we're just starting to dig into with this data as we're moving forward. And with that, we are wrapping up, guys. We have some other things that I mentioned that we're still working on. There's something called stable isotope analysis. Has anybody ever heard of this? Oh, good. So we took tissue samples galore, and one of the tissue samples was to conduct stable isotope analysis, and we literally just got the results back. So I was unable to dig into it in time for this talk. But what we try to do with stable isotope analysis is look at carbon and nitrogen levels in the tissues, and that can tell you the trophic position of the fish or how they fit into the food web. And the reason we want this for the dry tortugas is to then use this as a baseline for other parts of the keys. Are other animals with more intense fishing or different types of habitat degradation, so to say? Are these animals similar in trophic position to these pristine areas or these areas that have no fishing, or are they further away? If there's restoration efforts going on, do those restoration efforts move that needle for this trophic position to get them closer to the dry tortugas? Like I said, we're working with a graduate student for the genetics, and we're also using this baseline for other questions. There's a lot of information that is still not known. So once we have all of this movement behavior down, it would be very interesting to take that to these fished areas and do similar work and ask those questions again, like, is fishing influencing their home range size or their reproductive behavior during breeding season, things like that. And to wrap it all up, I just want to point out, like, this project was time intensive, and it took a lot of effort from a lot of different people to pull all of this off. And I could not be up here talking about this without acknowledging all of those partners, both FWC and at the Park Service, that helped to make this project happen. We have a lot of very very amazing people that are dedicated. It might not hurt that you get to work in the Dry Tortugas when you're working on this project, but it still took a tremendous amount of effort. And again, this is not the only thing that my team does. We also conduct acoustic telemetry on other animals, such as mutton snapper, gray snapper, grouper species. We conduct those aerial surveys, like I said, fish counts. We are talking to resource users, asking them their perceptions around closed areas and things like that, or fishing regulations. So there's a lot that we still do in addition. And with that, I would like to say, teamwork makes the dream work. Thank everybody who helped be a part of this, and especially the support from